So these are some concepts that we have learned. Today what are we going to learn is the basic structure of a prokaryotic and eukaryotic cells. Let me draw two cells here to understand the concept of cells. Say suppose I am talking about a particular cell. I am talking about a particular cell. Now this cell has a nucleus. Okay. And the nuclear region will have some hereditary material. So what are the parts inside this cell? What are the parts inside this cell? This cell, what are the parts inside? Bacteria. No, what are the parts inside this cell? This cell has got parts known as the nuclear membrane. This cell has got parts like the nuclear membrane. And what about the fine hair-like structures, thread-like structures known as chromatin fibers? These are called chromatin fibers. So what I told you about the concept of prokaryotic and eukaryotic is, if this is one type of a cell, then this is of a prokaryotic or a eukaryotic cell. This is an example of a eukaryotic cell. This is an example of a eukaryotic cell. What is the meaning of eukaryotic? I told you, U means advanced cell. In comparison, if I have to draw the structure of a bacterial cell, bacterial cells are not that big. This is just for an example I'm drawing. Say suppose now I talk about the structure of a bacterial cell. Now if I have to draw the structure of a bacterial cell, which would be something like this, and now they will be having some long hair-like structures for the process of movement. And if I now try to understand them, do they have chromatin? Yes, they also have got chromatin structures. But then this chromatin would be quite long and extensively coiled. And it is the same single chromatin that I'm talking about. They would have a single chromatin fiber. They would have a single chromatin fiber. So now that we are talking about the prokaryotic and the eukaryotic cells. So what I told you is, if this is a type of a cell that I'm drawing, then this type of a cell is known as prokaryotic, where pro means primitive. So then how do we understand that it is a primitive cell? Because this chromatin material is single, one in number, and it is extensively coiled, very thin, extensively coiled. But what about this side? The chromatin fibers are many. They are many in number, maybe 20, 30, 40, many in number. And they would be covered by a membrane. And what is the name of that membrane? Nuclear membrane. So in such type of a developed cell, we have got two distinct compartments of a cell. One is the cytoplasm, the other is the nucleus. Do we have anything here? No, there is nothing known as this. So if I have to give one name to this single chromatin fiber, then I should say this type of a nucleus is known as the nucleoid. This type of a nucleus is known as nucleoid. Meaning what? A nucleoid is such type of a structure in a cell where we have got only the hereditary material but no nuclear membranes around it. And another point we have studied the other day that we had discussed was that when we talk about the cell organelles, what are cell organelles? Parts of the cell which are going to perform many functions. We are going to draw the diagram now. So if I have to draw, yes, I will come up with there is one type of a cell organelle, there is another type of a cell organelle, there is something more, there is something more. But can I draw the same structures here? No, there will be no such type of a cell organelle in this type of cell. Most of them. In this type of a cell, hardly there will be some few proteins only. But in this type of a cell, there are almost about 8, 9 type of cell organelles. So these are some basic differences between prokaryotic and eukaryotic. Now let us learn how to understand the structure and the shape of the cells, especially now that we start the animal and the plant cells. Take out your pencils, take out your notebooks. First of all, we will draw the diagram, and then today we are going to learn the structure of the animal cell with the parts and the functions. Draw, start drawing. Take out your pencil, start drawing. Big enough. What should be the size of the diagram that you are going to draw? Half a page. The size of your diagram should be half a page. You will draw the diagram. How should you draw the diagram? Now that I draw the diagram, am I supposed to draw this diagram exactly in the middle of the page? No, I'm not supposed to draw the diagram in the middle of the page. This is my page here. Where am I supposed to draw the diagram? This is the margin, no? So I will draw the diagram here. I will not draw it exactly here. I will draw the diagram close to the left margin, keeping more space here to write the parts. Because I'm going to name the parts, no? So keeping more space towards the left. This is the diagram of the animal cell that we are drawing. We are drawing the diagram of the animal cell. Let us take this on the other side. Right here. Okay, so this is actually the structure of an animal cell that I'm showing you. This is what we have drawn. The structure of an animal cell. Now, what are we going to draw? The basic parts in the diagram. You are supposed to draw all the basic parts, the important structures that the cell is made up of. So, what are the important structures? Let's see. This is the animal cell. And what are the important parts? The cell will have two major regions. If you look into the diagram, stop drawing. I will give you some time later. Now listen to the explanation. So first of all, we are talking about this is the animal cell. Now in this animal cell, what are some of the important parts you are supposed to see? What are the nuclear regions? What is the nucleus made up of? You should be very aware about what are the parts that the nucleus possesses and what are the functions we are supposed to understand. So if you look into this diagram, this cell has got two important parts. The region outside the nucleus. What do we call the region outside the nucleus? There's one more part that we will realize. The region outside the nucleus, cytoplasm. Yes. The region outside the cytoplasm, we will call it as the region outside the nucleus, we will call it as cytoplasm. And the matrix inside the nucleus, what will we call the matrix inside the nucleus? Nucleoplasm. We will call it as nucleoplasm. And now that we talk about the nucleoplasm and the cytoplasm together forms the protoplasm, which is the living substance of a cell. Okay. So now, when we are talking about that, what are the important parts of the cell? How are you supposed to study? Now, one by one, we are going to understand what are the different parts. This is the nuclear region. Nuclear envelope is this. So now this becomes a prokaryotic or a eukaryotic cell. A eukaryotic cell, advanced cell. And then outside the cell, outside the nucleus, you get so many parts here. This is one, this is one. We name them separately, mitochondria, Golgi, lysosomes. This is known as a smooth endoplasmic reticulum. This is known as the rough endoplasmic reticulum, and centrioles, and etc. Then, what are the functions of each of them? Each of these parts has got a variety of functions, just like the organs of our body. The organs of our body perform different functions, so same like that. These different parts perform various different type of functions. And what are they called? These are all known as now cell organelles. We name them as cell organelles. So now we are going to study the function of each one of them and what are some of the detailed structures that we are supposed to understand. So let us come across the cell organelles one after the other. First of all, we are going to start with the cell membrane. We are going to start with the cell membrane. So that is the first organelle we are going to study, cell membrane. What are we supposed to understand about the cell membrane? What is the first point we are going to look into? If at all something about the composition we are to understand or the structure. So this is something that you will have to take a note for every cell organelle. The composition or the structure. The composition or the structure. That is the first point we are going to see. And what about the second? The second part that we are going to talk about would be the function, if at all any important function that they perform. If at all any important function that they perform. So these are the two heads that you have to study here. Composition of the plasma membrane. Now, if I magnify a small portion of the plasma membrane, we get to see that the plasma membrane is actually known to be made up of various different type of substances. Certain type of lipids, certain type of proteins are there. So when we are talking about the plasma membrane, so let us come across that the animal cell has got a plasma membrane which is made up of two major components. Which are the components? Let us see there. Which are the components? That means I'm saying that if I magnify a small portion of this, and then I try to see how does this structure look like. This structure more or less looks like this, with two basic layers of a fatty substance known as lipids. Two basic layers of fatty substance known as lipids, and in between there are some proteins there. In between there are some proteins like this, okay, which are quite broad enough, long enough to cover up the entire width, to span the entire width of the membrane, and some would be just partially present, meaning this entire thin layer that we get to see, if you magnify, we are going to come across that there are so many different type of proteins, and these proteins, some of them are arranged like this, some of them would be arranged at one particular region on the upper end, some of them on the lower end, some of them spanning the entire width. You can see a little bit on the upper end, and they are completely spanning the entire width to completely dig down into the cytoplasm also. This is a basic structure. This is a complete structure of the plasma membrane if we try to see in detail a little bit. Yes.
Vacuoles.
Vacuoles in a plant cell, generally not.
Like plant cells have big vacuoles in a.
In animal cell,
vacuoles are small, which is spread over the animal cell.
Vacuoles in animal cell, if you don't draw, that also is okay. But if you want to draw, it's optional. Vacuoles are very small actually in plant cells, no, very small and few in animal cells. And in animal cells, there are so many cells where vacuoles are altogether absent also. So there are certain animal cells only where vacuoles will be there, and in some absolutely absent also. So that's why vacuoles become optional. We can draw to show them, not a problem also if you don't show. But these are the important organelles that you have to show. Now, composition, what is the composition that we are going to write? What about the structure we are going to write? The plasma membrane is made up of, or the plasma membrane has a fluid mosaic structure. Plasma membrane has a fluid mosaic structure. What is the meaning of the fluid mosaic structure? When we talk about the fluid mosaic structure, what does that mean? Fluid mosaic structure means, say suppose some of you must have already seen those mosaic floors. Do you know what are mosaic floors? Mosaic floors, you know, no? Sometimes we go to certain hotels and all. Even in so many buildings, you will come across still the buildings which are a little bit old buildings. You will come across still today. They have got those mosaic floors where it is a white cemented floor, and you get to see those multicolored marble chips there. You know, these are known as mosaic floors. So how do they make those floors? So first of all, they will put a layer of cement, a white cement, and then they will throw those marble chips, and then they will put another layer of cement and polish. So there will be certain marble chips which are completely inside, which we don't get to see from the upper surface of the floor. We don't consider. Quite a number of those marble chips are completely inside. You cannot see from the top. So quite a number of the marble chips are here already inside. Will you be able to see from here? No, you cannot see them. Some of the marble chips will be like this, like the second one that I have drawn. Some of the marble chips will be completely innervate, spanning the entire width. Some you can see from this side also, some you can see from that side also. And some of the marble chips will be on the upper layer only. So if I talk about the mosaic floor, on the mosaic floor also I get to see that there are some magenta color, blue color, and brown color. Those marble chips are there. So the ones that I'm able to see are actually the ones like this or like this, which has got a portion of it on the upper layer of the cement. And those which are already innervated, already embedded, we're not able to see them at all. There is something like this, no? So that is the reason the structure of the plasma membrane is also termed as a fluid mosaic model. What is it made up of? It is made up of two layers of lipids, two layers of fats. Lipids are nothing but fats, and one layer of proteins and carbohydrates also. Carbohydrates are also there actually. And how are the carbohydrates associated? The carbohydrates will be associated like this, like chains here. They will be associated like chains here. Okay. So this is the entire structure of the plasma membrane. So when we are talking about the composition, the structure, we have to understand one specific feature here, that the plasma membrane is known to be semi-permeable. We'll learn certain properties regarding semi-permeability after some time. First of all, let us learn about the cell. The plasma membrane is semi-permeable. What is the meaning of semi-permeable, or what is the meaning of permeable? Do you know? Did you come across this word permeable? Permeable has got something to do with the transport.
And permeable is the it means when the cell allows allows
some molecules. When we are talking about permeability, always remember, the moment I am talking about permeable or permeability is a property of the cell which allows the process of movement possible. So I would say that yes, the cell could generally, all cells, allow the movement of certain molecules and allows the exit of certain molecules both way. No. So now that I talk about that, what is the meaning of semi-permeable? Put a stress on this word semi-permeable. What is the meaning of that? Semi-permeable means the ability of the plasma membrane to allow only selected molecules to enter or leave the cell. Only selected few molecules, not all molecules can enter also and leave the cell also. Now, there's a reason also why only selected molecules can enter, because every particular cell does not require all. Say suppose I understand a stomach cell. The stomach cell will only be taking in proteins which are necessary to make its enzymes. I talk about the skin cell. The skin cell will only be taking in substances, ions and proteins which is necessary for making the skin pigment melanin. It does not require anything, or it does not require all the minerals at all. It's not so. So that is the reason we say that this semi-permeable, the plasma membrane is semi-permeable. What are some of the functions of the plasma membrane to remember? It holds the cell contents. It holds the cell contents. So what is the meaning of that? The plasma membrane can very well hold the cell content. Now, how is the cell content? I am talking about the cytoplasm, which is something like a gel. No, it is not a solid structure at all. That without the membrane also it can hold. Without the membrane also it cannot. Without the membrane it cannot maintain its shape. And that's the reason this plasma membrane is generally associated. The plasma membrane can generally give a proper shape also to the cell. That's why, no. So if this cytoplasm would have been a solid structure, then was there any need of the cell membrane? Then there was hardly any need of the cell membrane.
Prokaryotic and eukaryotic cells would not be there.
At the same time in our body, every time the cytoplasm would have got mixed one with the other. So that does not happen, because one cell has got one boundary and the other cell has got its distinct boundaries are there. So this is one. And what is the other one? It is also, the cell membrane is also necessary or helping in the maintenance of the shape of the cell. And also protection. Protection from unwanted substances. Protection from unwanted substances. So when we are talking about the plasma membrane does not allow unwanted substances to enter into the cell, because quite a number of times those unwanted substances can bring about a damage to the cell and its function also. So what are some of the basic characteristics that you have to remember? Keep a check on this word. We'll have to talk about this word in detail later again in another topic. So this is about the cell membrane. Let us talk about the next organelle now. What is the next organelle we are going to talk about? The next organelle that we'll talk about, let us take the most important ones first. We talk about the mitochondria. Can you see the mitochondria in the figure?
Yes, ma'am.
Can you see the mitochondria? I'll show you the mitochondria in detail again. Let us learn the structure and then we will write down the points. How does the mitochondria look like? What are some of the basic structures of the mitochondria? Can you see here? This is how a mitochondria when cut in a section looks like. A particular cell will have definite numbers of mitochondria. Now, if I'm talking about the animal cell can have quite a number of such type of organelles. These organelles are more or less spherical structures. They are double walled. They have got two walls. See here, when cut them in a section, it is found that they have got two walls. But the outer wall is even. It is just smooth and even. What about the inner wall? The inner wall is folded. The inner wall is folded. See here, the inner wall is folded. So what are these folds called? These folds are called the cristae. These folds are called cristae. These are called cristae. And one single fold is called crista. And inside also, they have got the matrix. Cytoplasm is here inside also. One special property of this mitochondria is that this mitochondria also has got some ribbon-like structures known as the DNA in them also, apart from the nucleus. So we are already.
Can mitochondria be a nucleus?
No, mitochondria cannot be a nucleus, but mitochondria can regulate its own function. Mitochondria can regulate its own function because it has got its DNA. Can the other organelles regulate their own function apart from the mitochondria? No.
But then the head of the cell is nucleus. So
it's nucleus. Correct.
It controls everything. But then if the mitochondria has got a DNA, it can control itself, right? It can only control itself. It cannot control anything else in the cell. The mitochondria has got its own DNA. So the mitochondria can allow the process of division, its own growth. Mitochondria can control the formation of various different type of enzymes and proteins, its own functional proteins only.
Can the mitochondria do anything related with the Golgi or the lysosome or the endoplasmic reticulum? No. But the nucleus can do everything. The nucleus will always give instructions for the formation of proteins and enzymes, etc., by all the other organelles, what type of a function the cell should show, etc. So now, when we are talking about the mitochondria has got its own DNA. The mitochondria has got its own DNA. So now, if you have to look into certain features of the mitochondria, I want you to draw this structure also. Huh? First of all, you draw this structure. Draw that figure. Very, very simple. That is. Draw that figure. Pencil. That one. The mitochondria and the nucleus. You have to draw it separately also. Then we will learn properties about the mitochondria. Yes, did you write?
Yes.
If you want, you can write. Otherwise, points are there in the volume also. You may just tick from the volume. Now you draw the mitochondria. As I discuss the points, you may just roughly write so that later when you tally with the group, you are aware that you are studying all the points. Okay.
Now, what is F0 and F1 complexes?
Now see there. Now you don't write that way. You just write only F0 complexes. Now what happens is when I talk about the structure of the mitochondria, say suppose I'm looking into, we'll learn the functions also, so you will understand all these things. Now this is the mitochondria that I'm drawing, no? Outer membrane. And then this is the inner membrane that we draw. What are these folds known as? What are these folds known as? Cristae. Now, if you look at the cristae in detail, there are some small knob-like structures here. Huh? There are actually some small knob-like structures. If I draw this in detail, it is actually like this. Every cristae has some knob-like structures like this. So every cristae will have a variety of such type of structures. Now, what are these knob-like structures known as? These are known as the F0 particles. These are known as the F0 elements or F0 particles. What is happening in these F0 particles? This is the major site where the process of energy formation happens in the form of ATP. Mitochondria is always the powerhouse of cells. We have always studied, no? Mitochondria relates to the formation of energy molecule. Energy molecule in what form? ATP. So now you know that inside the mitochondria there are certain structures known as the cristae where the process of ATP formation happens. Now the process now is not important for you, but only the name and the site, only that much you will learn now. And then the mitochondrial DNA. Complete this diagram, then I will mention about the points here. Done?
Yes, ma'am.
School started?
Yes, ma'am.
Started. Did they start with science, biology?
Yes.
What chapter is okay?
Yes, ma'am.
I mean, chemistry started. Chemistry started. Biology now started. Physics started. And biology now started. Tomorrow's holiday, that's good. And in this week also you have so many holidays. No?
Yeah,
No?
30 years too. 30 years too.
This week no holiday? Friday is a holiday. He knows. See, you all are not well informed. Friday is a holiday again. Ambedkar Jayanti. Why your diagram is always micro mini size? Why do you draw so small? You will get zero in such type of diagrams if you draw. Come on, erase that and I'll give you 2 minutes to draw. I already told you, no, the diagram. This is your bio notebook. Six so nicely is drawing the diagram. You have to follow the instruction that the teacher gives, otherwise it will be the same thing. No, you come here, spend money, time, and ultimately you do the same in your exams. What instruction the teacher? I told you just now, every diagram should be half a page. So very smoothly you get the 2 marks, 3 marks in your diagram. 9th standard, 10th standard CBSE, you have enough diagrams to draw enough diagrams. So if you draw those type of miniature diagrams, your mark also will become miniature, no? अच्छा लिखोगे, अच्छा ड्रॉ करोगे, तभी तो जाके अच्छा मार्क मिलने वाला है. नीटनेस, प्रेजेंटेशन अभी 9th स्टैंडर्ड में आ गए हो. This you have to really focus on now. Neatness and presentation, if you know, I'll tell you straight away that we as teachers, when we are correcting 60, 70 papers at the same time, if your presentation is very good, कभी-कभार एक दो mistake भी hide हो जाता है. And if your presentation is very bad, maybe all your answers are right, but at the end we are human, no? We don't feel like correcting such type of paper. So it becomes a negative thing. Maybe the student has written all answers correct. So always focus on presentation little bit more. Done? देखो पेंसिल भी लेके आया पता नहीं कहाँ से. CBSE साइज है पेंसिल लिया. अब से बायो क्लास में हमेशा पेंसिल्स रखो पाउच में, हाँ? पेंसिल बॉक्स में पेंसिल इरेजर शार्पनर होना चाहिए.
इसने मेरी पेंसिल.
अच्छा इसने तेरा ले लिया. इसने भी?
नहीं मैंने इसका.
अच्छा. All of you will bring your own pencils, huh? Bio class में, physics class में, there will be so many diagrams to draw, and you will really practice drawing big diagrams. अभी भी तुमने erase करके ऐसा कुछ बड़ा नहीं draw किया, अभी भी तुमने छोटा ही draw किया. This is not the standard size of a diagram. Show your diagram to him. देखो, half a page I told you. Diagram should be clear and neat. हाँ? हाँ, drawing हो जाएगा. वो 2-3 बार practice करो हो जाएगा. But you should be very clear about the size because 2 marks रहता है, कभी 3 marks का दे in the diagram. You should get simple diagrams है ये सब. Full marks मिलना चाहिए इसमें. हाँ? पेपर में अगर 4 डायग्राम्स रहेगा एंड हर एक में अगर 2-2 मार्क रहेगा तो उसमें तो तुम्हारा आराम से 6-8 मार्क्स आराम से आ जाता है. Simple diagram at all. Now, sure. When we talk about the mitochondria, look into this. What are some of the structural features of the mitochondria that we have to understand? We are talking about structure. We have to understand about some of the features of the structure. Yes, the composition. Now, for the mitochondria, we are not talking about anything composition. We are only talking about the structure now. Mitochondria has double wall. It is double walled. Remember, it is double wall. And what is the other feature to remember? The inner wall is folded. The inner wall is folded. And what about the folds? The inner wall is folded. You'll have to remember this feature. And what about the folds known as? The folds are known as cristae. The folds are known as cristae. Cristae is plural when I'm talking about quite a number of them. So singular is crista. And what else? The cristae contain the F0 particles. The F0 particles. Which are what are F0 particles?
And where the process of protein synthesis takes place.
The site of ATP and protein synthesis. The site of ATP synthesis.
That's protein synthesis. ATP is formed as a product, right?
No. When you talk about the F0 particles are actually the sites for ATP production along with proteins. Site of ATP and protein synthesis. So this is what you are supposed to write down. So ये हो गया structure. Function, let us now try to understand. What is the function of the mitochondria?
Giving energy to the cell.
Producing energy or producing the energy currency. Producing the energy currency of the cell, that is ATP. And do you know the full form?
Adenosine triphosphate.
लिखो, bracket में लिखो. All of you should know this. This separately for 1 mark you get a question. Adenosine triphosphate. लिखो, adenosine triphosphate. Function, adenosine triphosphate. That is the function. So that is one function of mitochondria, producing the energy currency of the cell. Then mitochondria also brings about cellular oxidation. Mitochondria also brings about cellular oxidation. What is cellular oxidation? Breakdown of food. Breakdown of glucose. Breakdown of food to produce ATP. To produce ATP. To produce energy. Okay? So these are the functions of mitochondria that you'll have to remember. Now, since we are talking about mitochondria, we have to understand this feature. What is the special feature, distinctive feature of mitochondria? The mitochondria is semi-autonomous. The mitochondria is semi-autonomous.
And it is function itself too and on the orders of the nucleus.
No, it is semi-autonomous because it has got its own DNA.
Yes.
And hence it can regulate its own function. It is not fully dependent on the nucleus. Is it dependent on the nucleus? Partially.
Yes.
Partially it is dependent on the nucleus. Now say suppose today, are you completely dependent on your parents?
Yes, ma'am.
नहीं? नहीं लगता तुम्हें तुम parents पे completely dependent हो? If today your parents say that I'm not going to finance you anyhow, then what will happen to you? You are fully dependent. But now, maybe after 15 years, 10 years from now, 15 years from now, when you start earning, you become semi-autonomous. You can you can be sufficient for your own survival. But yes, we are always with our parents. We are always dependent on our parents. So now that time we are semi-autonomous. We are not fully autonomous. But आज burger खाने के लिए भी पापा से लेना पड़ेगा ना पैसा. So but maybe now when you start earning, maybe you can buy your own car also at the same time. So that is called semi-autonomous. We are not fully dependent at that time. So there will be another organelle when we study plant cells. That time also we will come across one organelle, that is the plastids. Plastids also is semi-autonomous. So now we are talking about the mitochondria. Now write one question. Complete this and write one question. Why is mitochondria known as a semi-autonomous organelle? Why is mitochondria? You will get questions. What are Christie? What does the mitochondria produce? Just separate one mark questions also you will get this way.
Semi-autonomous.
Organelle. Organelle का spelling क्या है?
ORGANELLELES.
Yeah, correct. LE singular, LES plural. क्यों तुम्हारा अभी कौन सा बेटर डेज और आने वाला है? हाँ? बेटर डेज अभी भी तुम वेट कर रहे हो, कौन सा बेटर डेज की वेट कर रहे हो? यही जो डेज जा रहा है कोई अच्छे दिन है? हाँ?
10th में नहीं गए वो अच्छा है.
क्यों 10th पे जाओगे तो अच्छा है, जितना जल्दी 10th 12 खत्म करो अच्छा है ना. 10th में
6 महीने में जो करना है
क्यों तो वो तो अच्छा है ना. 10th में syllabus is so interesting. CBSE में all over you study CBSE. The syllabus of 10th सबसे ज़्यादा interesting है. Even 9th syllabus is nothing interesting. 10th का syllabus interesting है, all subjects. You will love to study. You love to study? Who loves to study? No, like, do you like to study your subjects कि मम्मी पापा डांटते तो ही जाके पढ़ने बैठते हो? हाँ? फिर क्यों डरते हो? क्या डर है?
बोर्ड एग्जाम।
तो क्या है? बोर्ड एग्जाम ही तो है। इतने सालों से एग्जाम दे रहे हो फिर और एग्जाम से डर क्यों? It's nothing. It's not a big lion that you have to fight with. Okay? Why is mitochondria known as a semi-autonomous organelle? This is a 2-mark question. Why is mitochondria known as a semi-autonomous organelle? Likho. Likha question? Write down the question. Now let me tell you the answer. Mitochondria is known as a semi-autonomous organelle because mitochondria is known as a semi-autonomous organelle because it has its own DNA. Because it has its own DNA. It has its own DNA and can synthesize. Now you're coming across all these spellings for the first time. Synthesize. पता है? Synthesize. Synthesize मतलब formation, no?
Like photosynthesis we had.
हाँ. Which line you missed out, बोलो?
Organelle.
कौन सा organelle थी? What did I dictate?
First question and the answer.
The mitochondria is known as a semi-autonomous organelle because it has its own DNA known as mitochondrial DNA. That is how we write. It has its own DNA known as mitochondrial DNA, which carry information. What I said? Read out that sentence what I told you.
Because it has its own DNA and can
Synthesize variety of proteins. Synthesize variety of proteins and regulate. Synthesize variety of proteins and can regulate its own division and function. Can regulate its own division. Can regulate its own division and function, and is partially dependent on the nucleus. And is partially dependent on the nucleus. Partially dependent.
Why is mitochondria important?
Why is mitochondria known as semi-autonomous organelle?
And partially dependent on
Nucleus. You have completed the answer? Please take it from here. और तुम थोड़ा writing practice थोड़ा speed up करना पड़ेगा. Continue again in the next class. क्या? वो किसी में ठीक है